Hello, everyone. I wanted to thank Gary and the IDG Outreach members for the opportunity to present some of our work today. And so today I want to tell you a little bit about some of the work we've been doing as part of the IDG consortium, and particularly in both developing new tools for mining the dark kinases, as well as in using evolutionary context for formulating and testing new hypotheses on dark kinase functions. So particularly, I'm going to just focus on two recent studies. The first one is on characterizing the ULK4 dark kinases in collaboration with Stefan Knapp's group, and some of our ongoing work on developing the KinView framework and the protein kinase ontology framework for dark kinome mining and annotation. So this phylogenetic tree of the 524 human kinases really depicts the relationships between dark kinases and light kinases. So the dark kinases are in darker colors. And as you can see from this tree, that the dark kinases are evolutionarily related to many well-studied or light kinases. So the question is, how can we use the wealth of information on light kinases to predict or hypothesize about their underlying functions? And this is obviously a challenge because from our studies on light kinases, it's clear that within the kinase family, the challenge is not just to predict their substrates, but also to understand the unique modes of regulation in signaling pathways. As you can see here, for a subset of well-studied kinases, even though they all share a common catalytic scaffold, they each have evolved unique modes of regulating their catalytic activity. And understanding what these unique features are and how they contribute to unique modes of regulation is also critical in fully understanding the functions of dark kinases. So our hypothesis or our working premise for a number of years has been that these underlying mechanisms of unique modes of kinase regulation are encoded in primary sequences, and that by systematically comparing and contrasting evolutionarily related kinases, we can tease out those features that contribute to unique mode of regulation. And so here's an example of work that we did several years ago for the AGC family of kinases, where we delineated the evolutionary constraints that distinguish the AGC group of kinases from other evolutionarily related kinases. And what this revealed was some of the strongest AGC constraints are mapped to the kinase domain and actually tether a conserved C-terminal tail segment that wraps around the kinase core. And that finding provided a hypothesis as to how the C-terminal tail could function both as a cis regulator of catalytic activity, as well as serve as a trans-regulator in the sense that it could recruit other scaffolding proteins and uniquely tether these interaction sites on the C-tail to the kinase core. And as you would have heard from Alexandra Newton's talk yesterday, within PKC, how this sort of framework with the C-terminal tail as a cis and trans regulator provides a way of understanding unique modes of PKC functions. And we believe that several of the dark kinases within the AGC group may also be using a similar framework, which we can then begin to tease out using the distinguishing features that we've already identified for AGC kinases. Likewise, within the CMGC group of kinases, they all share certain features that uniquely tether a CMG specific insert segment to the rest of the catalytic core, and really provides a framework for then annotating or generating new hypotheses on some of the dark kinases, such as HipCase, which belong to the CMGC group. And that's the work we're doing or will be doing in collaboration with Natalia Jura at UCSF as part of the IDG RO3 awardees. We've also been working on delineating the features of various tyrosine kinases and the efferent receptor tyrosine kinases. And that knowledge will also be useful in understanding, although there are not that many dark kinases within the tyrosine kinase group, but there are some dark pseudo kinases that we're interested in studying as well within that. But for today's talk, I wanted to just focus on some of our recent work on this ULK4 pseudokinase, which was done in collaboration with Stefan Knapp. And because it's a dark kinase, there is very little information on the ULK4 kinase itself, and that is the reason why it's been classified as a dark kinase. But at least a subset of the papers that are available on ULK4 suggest that it plays a role in neuronal functions. In fact, ULK4 has been observed to be highly expressed in human and mouse brains, adult human and mouse brains. And it also has been implicated in microtubule functions through acetylation of alpha tubule. And there's some recent studies that suggest ULK4 as a prognostic marker for schizophrenia. And so we and others have previously shown that ULK4 diverges from other closely related kinases in key catalytic sites. So here's a sequence logo showing ULK4 in comparison with other closely related kinases or functional active kinases. And as you can see here, these are some of the hallmark catalytic motifs of kinases, and that ULK4 really diverges at some of these positions. So this is the key catalytic lysine that is involved in coordinating ATP in many functional kinases. But across different ULK4 orthologs, in particular in humans, the lysine is replaced by a leucine. In fact, other orthologs of ULK4 have a lysine at that position. I'll get back to that in a minute. But within human ULK4, that key catalytic lysine is significantly diverged in that it's replaced by a leucine. Likewise, this glutamate is also replaced by other hydrophobic amino acids, and you see divergence in other magnesium coordinating residues such as this DFG aspartate. A recent structure of the ULK4 kinase by Stefan Knapp, and also a previous structure from Mount Sinai, Michael Azara's group, showed that the key catalytic variations in the active site are actually predicted bioinformatically, contribute to a very divergent active site within ULK4, in that despite lacking some of the key catalytic features, ULK4 is able to bind ATP. And so this was really interesting, because if you compare ULK4 with the closely related ULK1, as you can see here, ULK1 has this key catalytic lysine that coordinates with the glutamate, and that salt bridge is critical in coordinating and keeping the ATP in position in many kinases. However, in ULK4, the lysine is replaced, as I said, with the leucine, and there is a tryptophan in place of the glutamate. So it was really intriguing as to how, despite these variations, ULK4 is able to bind ATP. So just to give some context, much of our understanding of how kinases bind to ATP have come from Susan's pioneering studies on protein kinase A, where she's shown elegantly that mutation of the lysine to even an arginine abrogates ATP binding and impairs catalytic activity. And in ULK4, as I said, it has a leucine, but still binds PPNP. And the only example of a kinase where you can still bind ATP in the absence of the lysine is with this Wing3, where the lysine actually migrates from the beta-3 strand to a beta-2 strand and still coordinates with the ATP in a manner analogous to pKa. So we were curious whether something like this was happening, where the lysine is kind of migrating away from the active site and coming from different parts of the protein. And so in the crystal structure, there is a lysine that comes from the C helix and coordinates with the beta and gamma phosphate of ATP. But surprisingly, when you mutate those lysine residues in the vicinity, this lysine and other lysine residues, surprisingly, there's no impact on the stability of the protein. And in fact, even in the presence and absence of the magnesium or in the present with ATP and ADP gamma of there is no difference in the thermal stability of the protein, suggesting that lysine is really not contributing to stabilization or binding of ATP as predicted. So this is when we started looking at the orthologs of ULK4 from diverse organisms. And what was interesting was that some of the plant and protist ULK4 seem to actually conserve some of the canonical active site residues like the functional counterparts. And that this divergence of the active site residues only selectively happened in mammalian ULK4. And in fact, what Safal in my group also observed when he did this broader phylogenetic analysis was that the loss of the catalytic site within mammalian and several chordates was strikingly correlated with this extended activation segment in these species. So in other words, if you look at mammalian ULK4, they have much longer or an extended activation segment that kind of wraps around and interacts with the C helix, while some of the plant versions, plant orthologs of ULK4 have a much shorter activation loop, which is of the more canonical length. And so one of the things we're doing right now is to figure out whether this extended activation segment has anything to do with this unique mode of binding ATP in ULK4. We're doing molecular dynamic simulations of ULK4. And interestingly, at least within the microsecond time scale, we're finding some charged residues, particularly lysine and arginines within this extended activation segment that seem to coordinate in ATP or during the course of simulation. And so one of the things we're trying to do is to figure out whether mutating these charged residues would have any impact on ATP binding, and whether that could be compensating for the loss of the canonical active site lysine. So while this is ongoing, Stefan Knapp has also been collaborating with Rob Rottapel and Brian Roth to identify the interaction partners of ULK4, and with many pseudokinases, because they have diverged in catalytic functions, they nevertheless maintain the non-catalytic or protein-protein interaction functions. And so, so Rob and Brian have been trying to identify the interaction partners of ULK4 using this bio-ID approach, where based on biotinylation of interacting proteins and subsequent isolation of these biotinylated proteins through mass spec, they've been able to really map the interaction partners of ULK4. And interestingly, some of the kinases, closely related kinases and phosphatases, have been identified as putative interaction partners, as well as several of the proteins in microtubule associated proteins. We have been trying to understand, in addition to these divergence in the active site, are there other selective constraints acting on ULK4 that would provide us insights into which regions of the kinase domain could be involved in these protein-protein interactions. And we've identified some selective ULK4 specific constraints on the N-lobe of the kinase, in fact, on the N-cap region of the kinase, where ULK4 significantly diverges from other closely related ULKs. And so our hypothesis is that perhaps these constrained residues on the N-lobe are presumably involved in protein-protein interactions. And again, this is ongoing work where we're trying to really tease out these interaction interfaces or functional sites within the ULK4 kinase domain. So this work illustrates how we can use the evolutionary constraints acting on kinase sequences in conjunction with experimental studies to generate mechanistic hypotheses or insights into some of the dark kinase functions, such as ULK4. And so now I want to shift gears and tell you about some of the informatic tools we're developing to further accelerate the functional characterization of these dark kinases. And so as I've shown in the previous example, this sort of comparative analysis is a conceptual starting point for generating and testing new hypotheses on dark kinases. And so to enable the community to do these sorts of comparisons in a more effective and easy way, we've been developing this tool called KinView that allows a comparative analysis of dark kinases and light kinases. And so we've curated multiple sequence alignments of various dark kinases and light kinases, and these alignments are now placed in the context of this evolutionary hierarchy. So using this KinView tool, you can essentially navigate these alignments, curated alignments, using this evolutionary hierarchy that everybody's familiar with. So for example, if you wanted to really compare the different ULK4s, you could select the ULKs you're interested in. And if you wanted to compare the 4 ULKs, you could do that, and it displays the alignments, and you can actually move the alignments in whatever order you want. And then you can select on the mutations that have been identified and mapped to each of the aligned positions. And you can also change the display of the residue numberings. For example, if you wanted to compare the ULK with the pKa numbering, you could do that by selecting the corresponding reference position. So there are a lot of interesting utilities like this that could help in navigating the evolutionary data that is encoded in these sequences, and using this sort of comparative framework for then generating new hypotheses. So we've also placed the secondary structural information on top of the alignment, so you can also map these patterns onto secondary structures, and from there onto 3D structures. So this is a tool that is being beta tested, and it's available for use for the community, and you can download it from this ProKino link that is shown here. So please use this, and if you have any comments or suggestions, feel free to email us or contact us. And so we've developed this tool in a way that this could be extended to any protein family of interest. And in fact, we're working with the IDG consortium to extend these tools for GPCRs and ion channels. So finally, in the last few minutes, I wanted to tell you about some of the other tools we're developing to address the data integration challenge in the kinase field. So as I've shown you, there is a lot of information on the molecular functions of various light kinases, and now with the IDG consortium on many dark kinases. And so the challenge is, how can we really integrate these molecular level data with the cellular data that's coming from the IDG consortium, such as the pathways that these kinases are involved in, the network context of the kinases, the phosphorylation, and of course the disease context. So because these information are currently stored in disparate sources and formats, and in fact several of the molecular level information is stored in unstructured text format in the literature, one of our focus for the past several years has been in building this knowledge graph, or an integrated knowledge graph that really links molecular level and cellular level data on kinases in human and machine readable format. So within the ontology or the protein kinase ontology, which we view as a knowledge graph, this really links major concepts such as sequence, disease, proteins, and pathways, and it as sort of classes, and it explicitly captures the relationships between these different concepts or classes. For example, a protein is associated with the disease, and this particular protein participates in a pathway, and this pathway has a reaction, and so on and so forth. So each of these classes are now instantiated with data. So we end up having a populated and a complete knowledge graph of the protein kinase knowledge base that is conceptualized and integrated using this ontology schema. So obviously our knowledge of kinases continue to expand as we characterize new light kinases and dark kinases. So it's certainly work in progress. But the current version of the ontology as is available for download and used by the community. And so the latest version of the ontology has about 837 classes, 82 data properties, and it's instantiated with data. So there's a lot of individuals. And so the current size of the ontology is about 3 gigabytes, and one can download and use the ontology. So a couple of applications, sort of common applications of the ontology, and one of the applications is using the ontology to find new associations between dark kinases and pathways using machine learning on knowledge graphs. And this is an ongoing work that we're doing. And unfortunately, I won't have time to tell you about that. But the other application of ProKino is in the generation of new hypotheses through integrative knowledge graph mining. And this is something that we and others have done, applied ProKino extensively for. And as illustrated here through these figures, one of the applications of ProKino is you could query the ontology for aggregate information linking multiple forms of data, such as an aggregate query that requests for all the mutations mapping to various kinases and their evolutionary hierarchy. And you can generate these sorts of graphical outputs that really links all these 3 different data types. Could also query for mutations mapping to different structural regions of the dark kinase, and you could get these sorts of images or figures that could then provide the database that the data that you need to then generate new hypotheses. So we've done this with multiple examples in collaboration with various researchers, and those are illustrated in these publications. And finally, we're working to integrate the ProKino annotations with PHAROS knowledge base that the IDG consortium is developing, and we're also working with the dark kinase knowledge base to integrate information from ProKino and link ProKino related information with these IDG generated resources as well. So in summary, I've given you a brief overview of how we're using evolutionary context to formulate and test new hypotheses on dark kinases using ULK4 as an example. I've given you a brief update on our progress with the KinView and the protein kinase ontology. So finally, I want to thank all the people who've been involved in this work. So Safal in my lab, in collaboration with Stefan Knapp, did the ULK4 work, which is just recently published in Structure. And the protein kinase ontology has been developed in collaboration with Chris Koshut at UGA, with several of the students here listed that are working with me and Chris on further developing the ontology and the visualization tools. So we're also working with Patrick Ayers and colleagues on other characterizing other dark kinases and pseudokinases, and several of the IDG RO3 awardees on the kinases that are listed here. So we've been also very fortunate in working with the large consortium, the IDG consortium, and we're finally very grateful for the funding from the IDG for doing the work that I've just described. So thank you for your attention, and I will be happy to take any questions you may have. And I wanted to thank you all again for your attention.